Bismillahirrahmanirrahim. My name is Dr. Sagheer Ahmad, and today I'm with you people with a new lecture that is about Krebs cycle. It is also known as citric acid cycle and tricarboxylic acid cycle. Why do we call it citric acid cycle? Because the first molecule which is formed in all these reactions, that is what this citrate, okay, or it is also known as citric acid. What? Citric acid. That's why we call it citric acid cycle. Students, today I will try to make it clear reaction by reaction, so that you may not cram it. The cramming is not learning. If you say what purifra, sealant, tetrapyrity, almanthizas, calmanthizanilate, arthropoda, canonameta, hemicordata, what is that? You must know. Why do we call the protozoa as protozoa? Why do we call purifera as purifera? There should be certain reason. Why you give certain name, taxonomic name? There is certain reason behind that. And here, if these reactions they happen to occur, there are certain reasons, okay, and you must trace those reasons, and you will learn by heart. Those reactions will be inscribed in your mind. Why do we call it Krebs cycle? Because all these reactions, these were first of all discovered by Sir Hans Krebs. Okay. As far as the site of these reactions is concerned, these reactions, all these reactions, they occur in the matrix of mitochondria. So I would like to suggest to you people that you must keep in your mind that if you are studying all these reactions, you suppose that you, along with me, are standing in the matrix of mitochondria, not in the cytoplasm, not in the cytosol. You are in the matrix of mitochondria. All these reactions, they occur in the matrix of mitochondria. In certain lecture, I've made you people understand how reaction by reaction glycolysis is completed, and at the end, what is formed? At the end, pyruvate is formed. This is what? Pyruvate. Okay? And from here, the story starts. Pyruvate is converted into what? This very compound, this very molecule, and that is what? Acetyl-CoA. You know, this is what? Acetyl group, and this is what? This one is what? Coenzyme A. We suppose that the coenzyme A is such a large molecule, and it's this very part, sulfur, that reacts with its hydrogen. And what happens? What happens? Here, you see, carbon dioxide is removed. This very COO. This carbon dioxide is removed. Okay? And this is what? S-CoA. What is left? S-CoA. Where has gone this hydrogen? I would like to tell you people where this hydrogen has gone. You know, this is in black color. This is that hydrogen. And where has this hydrogen come from? This hydrogen. This is that one. okay, from the carboxylic part of what pyruvate. This hydrogen is here, okay. So nicotinamide adenine dinucleotide is reduced. The oxidized form of nicotinamide adenine dinucleotide is reduced, okay. And what is formed? Acetyl CoA. This is what? Acetyl CoA. Acetyl CoA. This acetyl CoA is the molecule which reacts with another molecule, that is what? Oxaloacetic acid. What? Oxaloacetic acid. It is already present in the matrix of mitochondria. This oxaloacetic acid, it would react how? I would like to show you people the essence of this reaction. You see, this is what CH, and this is what COOH. I mean, as such, it is present here in what citrate. Here is what? Carbon. This one. Okay. This oxygen is here, right? Here is the carboxylic group already present in what? Oxaloacetic acid. I told you people, this is the first reaction. This is the first reaction, okay? And where is this something? What is its participation? This CH3 is, here is this carbon, and CH2. Where has gone one hydrogen? That is what? From the color pattern you can see, here is that hydrogen. Coenzyme A is released from here, okay, and it brings one hydrogen with it, okay, and that is what? Where has that hydrogen come from? From here, and it is now CH, and this carbon is this one, this oxygen is this one. Now, question is, where has this OH come from? And where has this hydrogen come from? This OH is this one, from water. And this H is this one. Okay, so you can understand that the water molecule enters into this reaction, and coenzyme A release, and acetyl part becomes the part of citrate, okay, or citric acid, and this reaction is completed. I mean, oxaloacetic acid and acetyl CoA, these two molecules react along with hydrogen and form what? Citrate. Next reaction, or the second reaction, is what? In this reaction, in the second reaction, what happens? Cis aconitase. This enzyme releases hydrogen from citrate. You see, hydrogen oxide, I mean, water is released. Hydrogen and hydroxyl group, I mean, water is released. And what is formed? Cis aconitic acid. This is what? Cis aconitic acid. This one. This is what? Cis aconitic acid. Cis aconitic acid undergoes the next reaction, which is the third one, you can say, and water enters, and what is formed? That is what? Isocitric acid. What this is? You see, citric acid. This is isocitric. What is the reaction? You can make notice of it. Here is what? 1, 2, 3. 1, 2, 3, 4. Hydroxyl group is at the third carbon. Here, hydroxyl group is at the second carbon. You can make the difference of it. Okay, and now let's trace where the hydroxyl group has attached and where hydrogen has attached itself with a cis-aconitic acid. This is hydrogen, you see, and this is hydroxyl group. Okay, now if it is clear, let's move to the fourth reaction. In fourth reaction, this is fourth reaction. Here you see isosociated dehydrogenase. This is the enzyme, and you know what are the meanings of dehydrogenation? I mean, hydrogen is removed. Okay, we have to trace now from where hydrogen are removed and where those hydrogen go. You see here, I have written in red color. There is certain reason. Okay, from here, this hydrogen is removed, and that becomes a part of nicotinamide adenine dinucleotide. It is what this coenzyme is reduced. And where has come this hydrogen from? From here. And you will see what here is double bond. This is same. Okay, and this carbon dioxide is removed from here. This carbon dioxide is removed. Okay, clear? Now this hydrogen is where? This is that hydrogen, black, and this is, you see, we have just supposed, otherwise hydrogen is never black or blue or red. Okay, I have just tried to give you people the concepts. Here, what is formed? This is alpha ketoglutarate acid. Alpha ketoglutarate acid is formed, and from alpha ketoglutarate acid, the fifth reaction starts, and here enters what? Again enters what? Coenzyme A, and nicotinamide adenine dinucleotide is converted into reduced form of nicotinamide adenine dinucleotide. Now let us try to trace the hydrogen. Where has this red hydrogen come from? From here. Okay, where has this hydrogen come from? It has come from here. Okay, and where has gone this very COO? This COO has been released as carbon dioxide. Okay, now where is SCOA? SCOA. This is acetyl. This is what coenzyme A. Okay, and you have stated that acetyl coenzyme A, when there is acetyl group attached with coenzyme A, we call it what? Acetyl CoA. When there is succinyl group, this is what? Succinyl group attached with the coenzyme A, we call it what? Succinyl CoA. This is what? Succinyl CoA. I would like to write its name. Succinyl CoA. This is what? Alpha ketoglutarate acid. Okay. This is what? Isocitric acid. This is what? Cis-aconitic acid. Cis-aconitic acid. Okay. This is what? Citrate. So let us now come back. Succinyl CoA, it undergoes the sixth reaction of Krebs cycle. And what is formed? Coenzyme A is released. This is that coenzyme A, which I have shown in the black color. This is coenzyme A. Okay, and where has this hydrogen come from? I would like to tell you people, we have to trace where this hydrogen has come from. Okay, now let's see the next reaction. This is same. Okay, and this CH2 is same. Okay, this CO is this one. This CO is here. Okay, and what? This is that one. And now we have to find where has this OH come from. This OH. Okay, and where has this H come from? Obviously, water has entered into this reaction. Water has entered into this reaction, and now I have to show this CO in the red color, and this is again in red color. Now I think the reaction is clear. Now this molecule is known as what? Succinic acid. What? Succinate acid. It is also known as succinate. When it participates in the reaction, it is known as succinate. When it is stable, it is known as succinic acid. Okay, now succinate or succinic acid undergoes the further seventh reaction, and what is formed? Two hydrogen are removed. Two hydrogen. This is CH2. This is CH2. One hydrogen from here and one hydrogen from here. These are removed, and these two hydrogen, these can be shown here. Okay, I mean flavin adenine dinucleotide, which you have stated already in my lecture, how FAD becomes FADH2. I told you people in certain in this very channel, you can search. Okay, in this reaction, two hydrogen, I mean from here and from here, two hydrogen are removed, and succinate is converted into the next molecule, and that is what? Fumarate or fumaric acid. This is what? Fumaric acid. Fumaric acid. Okay, fumaric acid undergoes the further reaction, and that is that you can number it as the 8th reaction. In this reaction, what happens? You see, it seems that two hydrogen have entered and one oxygen. That is what? Water. Okay, and you can locate here. Here, water has entered into this reaction. This is OH. Okay, and here one hydrogen. I mean, you can show this hydrogen here, which you show as what? CH2, and I think you have located these two hydrogen, and this is what? OH. In the next reaction, in the last reaction of Krebs cycle, you find what? Another reduction. That is what? Nicotinamide adenine dinucleotide in the oxidized form is there, and that is converted into what? Reduced form of nicotinamide adenine dinucleotide, and what is formed? You see that very place you have reached where what is formed? Again, oxaloacetic acid. Oxaloacetic acid is formed. Oxaloacetate is formed, and by that the Krebs cycle is completed. Okay, if you have understood well, I would like to suggest you people that I would like to request you people that you must subscribe my channel, okay, so that I may keep on presenting to people the new videos. Thank you.